All right. Can you see the full screen and hear me well?
Yeah, it's good.
Awesome. All right. Okay. Nice to meet you all, and thank you for having me here. And it's really my honor to kick off this symposium. And today I will talk about how to build foundation models for single-cell omics and imaging. So before we start, I want to say that all the work I will talk about in this talk are still not published. They are still ongoing work, but I put them on preprints, and also all the code, data, weights are available for everyone to access if you are interested. And specifically, I will talk about two foundation models. SCGPT is a foundation model for single cell, and MedSam is a foundation model for imaging. We definitely welcome your constructive comments and feedback. So what are foundation models? It is a very popular term, particularly post-ChatGPT. And you probably heard a lot from social media, particularly Twitter. And from academic perspective, really the term of foundation model is first coined from a seminal paper, which I show on the left of the screen. And it is co-authored by many Stanford researchers. And in this paper, they coined the term foundation model. And really the idea of foundation model is that you can pre-train this foundation model using massive amount of unlabeled data, such as text, images, speech from internet. And once this foundation model is trained, you can take this foundation model and adapt it to many downstream tasks with zero-shot or few-shot learning. So this is really the core idea of foundation model. However, this paper has 300 pages, so too long, don't read. And I summarize some key features for building a good foundation model as A, B, C, D. Here, A, algorithms. Most foundation model is built upon transformer-based deep learning architecture, which I will briefly talk about later. B, business or applications. The idea, as I mentioned before, of foundation model is that one foundation model can enable multiple tasks by transfer learning. And C, compute. Many foundation models require C, data. Data is the new oil for AI, and to build a foundation model, it usually requires massive amount of data for pre-training. And as in single cell, we ask ourselves the question, can we build a foundation model for single cell data? The motivation is quite clear to us. While texts are made of words, cells can be characterized by genes. And this analogy really inspires to explore foundation models for single cell. Certainly in the long run, we expect such model has the potential to gain general understanding of the underlying biology. Here, I'm not claiming that language and biology have the same mechanism, but there are enough technical similarities really inspire us to explore these directions. And back then, we really started this project, SCGPT, even before ChatGPT was released. And back then, we didn't know whether it's a good idea or it's too crazy. And let's look at these four key features, A, B, C, D, I mentioned before. So A, algorithms, really, as I mentioned, all the foundation model are based on transformer architecture, which was first proposed in a seminal paper in 2018 by Google titled Attention is All You Need. And in this paper, a new deep learning architecture called transformer is proposed. And if you look at the architecture of transformers, it basically consists of two parts, an encoder and a decoder. An encoder will take the input and map it to a latent space, and the decoder will take the latent space and trying to decode out the output. And if you stack enough layers of encoder, you will get a bird-like model, which is again invented by Google. And this was one of the first prototype of large language models. And in single cell domains, we also have similar architect, which is basically encoder-only architect, such as SCBird scFarmer, which is a model from our lab too, and GeneFarmer, which was recently published in Nature from Harvard. And if you stack enough decoder-like model, you will get GPT-like model, which was first invented by OpenAI and ChatGPT-1 until ChatGPT-2, ChatGPT-3, and recent ChatGPT-4. They all fall kind of the same type of models. And this model is really the same model we use to build our scGPT, a GPT-like model for single cells. So we have the algorithms. Let's look at the bee business. There is really no shortage of business in single cells, such as cell type clustering, batch integrations, multi-omic integrations, cell type annotations, gene regulatory network, or perturbation predictions. Certainly, there are many other tasks we can explore in single cell. However, in our scGPT paper, we mostly focus on these 6 applications. C, computing, there's not much to say here. We were lucky to have some GPUs from Vector Institute, which is a AI hub in Canada. And certainly it's still far from being enough. And I'm sure this is a constant struggle for many academic labs nowadays. Let's look at D data. And we were extremely fortunate to have worked with Human Cell Atlas, which is really a centralized resource to curate a massive amount of single-cell RNA-seq data. And specifically, we worked with cell-by-gene census, which helped us curate more than 30 million human cells, covering more than 500 different data sets. And if you look at the major tissues, they are heart, blood, brain, lung, and etc. So now, after going through these ABCD components, we were more or less confident that it is a good idea to start training scGPT, a foundation model for single cell. That's exactly what we did for the past few months. So specifically, scGPT consists of 2 steps. First, generative pre-training, and second, fine-tuning for downstream tasks. And let's look at these 2 steps one by one. First, generative pre-training. As I mentioned, we took 33 million human normal cells from Human Cell Atlas, specifically cell-by-gene census, and then covering major tissues such as brain, blood, lung, heart, etc. And it took us about 20 A100 GPUs for 2 weeks. So it is really a heavy lifting in terms of computing resources. And after 2 weeks, we have a good model, and the loss function works just fine, but we didn't know what we get. So we didn't know how the model was trained. So what we did for sanity check is that we randomly sampled 30,000 cells and plot UMAP, which I shown here. And also we kind of using the labels from cell by gene just as a sanity check. And we see clear cell type ontology hierarchies. So this kind of give us confidence that this model is well trained. If we dive deeper into some of the technical details, the input for scGPT contains 3 sets of tokens. First, the gene identifiers. Basically, we just tokenize the gene names. And second is gene expression values. What we did was quite simple. We simply bin the gene expression values and then tokenize the resulted binnings. The third input for scGPT, we call it condition tokens. It's an innovation we made to being flexible to incorporate some metadata. For example, we can use these condition tokens to incorporate which genes are perturbed for perturbed sake. So this condition token is really a very flexible mechanism for us to incorporate metadata for single cells. And once we have these 3 sets of inputs and we tokenize them, we add them up as the input to multiple layers of transformers. As I mentioned, the algorithm behind most of foundation models are basically transformer blocks. And same for our scGPT, we have multiple layers of transformer blocks. And specifically, since we are generative pre-training, the way we use generative pre-training scGPT is really an innovation we made for masked multi-head attentions. However, in languages, the order of words matters. For example, I like coffee is different from coffee like I, right? The order matters a lot to understand languages. However, it's not, the order of genes does not matter that much in single cells. So in order to adapt this autoregressive training to SCGBT, the innovation we made is that we use this cell embedding CLS token in the attention modules, and we simply rank the genes based on the attention values to this particular cell. And based on these attention values, we kind of know how important each gene is with respect to particular cells. And then we create this manual order of genes, and then we can chunk them into different chunks, and then just use the same autoregressive training ChatGPT used to train languages to train SCGBT in an autoregressive way. Certainly, there's lots of technical nuance, which unfortunately I don't have time to talk about. For example, we use teacher forcing in the beginning epochs of the training just to make sure the model have some good values to start in generations. There are lots of other technical nuance, such as how to select batch size, how to select learning rates. Unfortunately, we don't have time to cover this in more details, but if you are interested in knowing these technical details, feel free to check out supplementary material in SCGBT. So as I mentioned, once after 2 weeks of generative pre-training, we have this bulk foundation model. And the second step is really perform fine-tuning for downstream tasks. The first task we have for SCGBT is to integrate multiple batches of RNA-seq, which is a common task in single-cell analysis. And really, the key for fine-tuning is to define your loss functions. And specifically for multi-batch integration, we define a loss function consists of 2 terms of losses. The first term of loss is contrastive loss. The idea is quite simple, and which is commonly used in deep learning, is that if 2 cells are neighbors in the original gene expression space, they should be neighbors too in the embedding space. And the second terms of loss for multi-batch integration is adversarial loss, which is a loss we obtain from a previous GAN approach. The idea is, again, very simple. In order to remove batch effects, we basically want SCGBT fail to recognize which batch the single cell come from. And once we have defined this loss function, the rest of fine-tuning is quite streamlined. You can simply calculate the gradient and perform backprop with SGD, for examples. And in order to really verify efficacy of this fine-tuning, we took PBMC 10K cells, which is kind of human blood cells, and then we perform the fine-tuning, and we compare with specialist approaches such as Seurat v3, SCVI, and Harmony, and we reported average bio as a metric to evaluate the goodness of this model. And if you see the value and the UMAP, you will find that SCGBT, by simple fine-tuning, outperform these specialist approaches. And we then go ahead and tested multi-omic integration, where we have not only single-cell RNA-seq, but also single-cell ATAC-seq. And I just want to emphasize that we actually don't have a foundation model for ATAC-seq due to data limitations. What we did is simply map the peaks into genes and then use the same pipeline of fine-tuning multi-batch integration of RNA-seq for multi-omic integration. And what we did is following. We took the 10x multi-omic PBMC data and then compared with Seurat v4 and SC-GLU, and again reported average bio, and you will find that similarly, SCGBT outperform specialist approach such as Seurat v4 and SC-GLU, which are specifically designed for multi-omic integration. We also tested a more challenging task for multi-omic integration. We call it mosaic integration. Mosaic integration is a more challenging task because not only we need to integrate RNA-seq and ATAC-seq, we also integrate protein omics. So this slide is talking about how we can perform fine-tuning for multi-omic integration. What we did is that we collect human PBMC cells consist of single-cell RNA-seq, single-cell ATAC-seq, and single-cell protein omic for unmatched cells. Again, we compare with SCMOMAT, which is a specialist approach that is specifically designed for mosaic integration. And similarly, we find that SCGPT outperform SCMOMAT in multi-omic integrations. And by far, you kind of see the pattern that SCGPT as a foundation model, the same model with proper fine-tuning, it can outperform many specialist approaches across many tasks. We further tested cell type annotation by looking at 3 data sets, one of which is cancer, 2 others are immune cells, by reporting F1 values, accuracy, and again, SCGPT outperform 2 other specialist approach in cell type annotations. And people ask, cell type kind of supervised fine-tuning, can you do zero shot? Meaning you don't do any fine-tuning, you just simply run inference on some new cells. So we test the ability of zero shot for SCGPT by running SCGPT on the COVID-19 data. The reason we chose COVID data is that during pre-training, we never see any COVID data. We want to test how generalizable SCGPT is for zero shot. And we find that by simple reference mapping, zero shot can actually generate very good results on COVID data. And this gives us lots of confidence to continue work with Human Cell Atlas to provide this reference mapping functionality for large cell atlas, because we have so many single cell. Yeah, no worries. This slide says we work with cell by gene to provide this functionality of zero-shot reference mapping from a large cell atlas. The steps to generate this reference mapping is quite simple. First, we generate zero-shot cell embeddings through foundation models such as SCGPT, and then we train and store search index through these embeddings, and then when new cells comes in, we simply run retrieval and propagate labels on the new data. And this functionality now is provided through Human Cell Atlas, and the storage cost is extremely small, less than 1 gigabyte, and the query speed is extremely fast. That taking a new data set of 4,000 query cells, it costs less than 10 seconds on CPU, less than 1 second on GPUs. And we also find interesting scaling law of foundation models. The experiments we did is that we randomly sample 30,000, 300,000, and 3 million cells, and then we train exactly the same SCGPT, and then we report the accuracy on cell type assignment. And the curve on pink shows the scaling law. And this curve gives us 2 observations. First, unsurprisingly, the more cells you have during pre-training, the more capacity you will have on the foundation model. So this is not surprising. We often see this in natural language processing, in computer vision as well. The second observation, which is a little bit interesting to us, is that once you surpass 3 million cells for pre-training, the performance gains start to diminish. The performance still increases, but the gains start to diminish. And this really gives us lots of insights about how many cells we need, what is the best way to curate the single cell data for pre-training. We also tested on gene perturbation response. The idea is similarly, we collected multiple benchmark data set on gene perturbation, and then we predict the gene expression after perturbation and report the correlations. And similarly, SCGPT outperforms specialist approaches such as GEAR, which are specially designed for perturbation predictions. So this really shows the generalization ability of SCGPT on multiple tasks. Once we published this model, SCGPT, we got really huge tractions from the community, and many wet lab experiment people kind of reach out saying, well, the model looks fantastic, but we don't have GPUs. How do we access, how do we run your SCGPT? And in order to make sure SCGPT is accessible to everyone, we worked with AWS, which is a cloud provider, to create this SCGPT hub. The way SCGPT hub is that as a wet lab experiment biologist, you can sign up first, and then you log in, you upload your own single cell data, you choose which foundation model to start, and then you set up some hyperparameters, and then you submit your job. And then you just go out and have your coffee, and then once you finish your coffee in 3 to 5 minutes, you come back and you have all the wonderful embeddings from SCGPT, including all the visualizations. And we only publish these portals in 3 months, and already we have thousands of users. We talk about a lot of interesting properties of SCGPT. However, there are many limitations for SCGPT. First, SCGPT is mostly limited in RNA-seq. Despite that SCGPT is capable of integrating multi-omic data, including ATAC-seq, we really only have a well-trained base model for single-cell RNA-seq due to data limitations. The second limitation is that SCGPT, despite of learning meaningful gene programs, is still limited in causal inference. In other words, it's scaled by associations. The third limitation is that the pre-training of SCGPT is very compute intensive. However, we did the heavy lifting for you, and we share the model and the weights with everyone. So now the fine-tuning is extremely lightweighted. Most of the time, for any normal single-cell data, fine-tuning, it only costs like 3 to 5 minutes. Last but not least, we still think zero-shot capacity of SCGPT is limited. Although we test that zero-shot capacity in reference mapping is quite impressive, but zero-shot for many other tasks are still limited. In other words, we don't think SCGPT is a GPT-4 moment for single cell. Instead, we think SCGPT is a GPT-2 moment for single cell, where we show some potentials, but it's far from perfect. There's still a long way to go to really show the power of foundation models for SCGPT. And the purpose of this foundation model, particularly like SCGPT, is we want to see some paradigm shift. Particularly, we want to move from hypothesis-driven approach to data-driven approach. And many current practice rely small center, single center validations. And with SCGPT and the Human Cell Atlas, we want to move to large-scale multi-center validations with kind of general use framework with end-to-end hardware such as GPUs. In order to achieve such paradigm shift, the collaboration between biologists and computer scientists is really essential. And a natural question we can ask is, can we extend SCGPT to spatial omic? Really, the challenge is data scarcity. We simply don't have too many spatial omics available to train a foundation model for spatial omic. And also, spatial omics sometimes involve biology images. And we really don't have a foundation model for biological images. However, I think it's very possible with the current pace of foundation model development for biological images. We were very confident that we will see a foundation model for biological images in 2 to 3 images. We published this commentary in Nature Method. And also our lab has spent significant time to work on these directions. And recently we published a paper in NERPS that we call BLEEP. Basically, it's a transformer-based approach that can take the HNE images in 10x VZM single-cell data, and then from these HNE images, we can predict the gene expression values. And we compare with some other traditional methods, such as linear model and convolutional neural network-based approaches. Really, we find that BLEEP is the only model that can generate decent correlations from HNE images and the gene expression values. However, the accuracy is still not satisfactory. In order to have these community efforts to start to develop this foundation model for biological images, in 2022 at NERPS, which is the largest AI conference, we organized a challenge to provide biological images and invite computer scientists from all over the world to work together and curate this foundation model for biological images. And specifically, we collected a very diverse data set of biological images covering 50 different biological experiments with 4 different microscope types. And overall, in training data set, we have 200,000 cells, and we hold out 60,000 cells for testing. And we had received overwhelming response in participation from all over the world, and we summarize all the results of this challenge in this archive paper. So if you're interested, feel free to dive deeper. But in a high level, through this challenge, we find that the winning solution is actually based on transformers, and it can outperform UNet type of model, which are commonly used in current biological image segmentations. So this is a good news. However, we tested the generalization ability of this winning solution. It's still not satisfactory, and we don't think it is a foundation model yet. And then the idea we had is, can we borrow something from medical imaging? We actually have a foundation model for medical imaging because we have lots of medical image data. And specifically, the model I want to talk about using the last few minutes is that is a model called segment anything in medical images. We call it MedSam. And what we did to develop this MedSam is that we took the encoder from segment anything module, which are published from Meta, and then we continuously pre-training this image encoder using 1 million medical images. And this 1 million images covering a variety of medical images such as MRI, CT scan, ultrasound, X-ray, pathology, you name it. All the medical images you can find were used in this pre-training data set. And once the model is well trained, we find that MedSam can outperform many specialist approaches such as UNet, DeepLab v3, and show great results in some internal data sets. We also validated MedSam on some external data set, and again, we see superior results in comparison to UNet and DeepLab. And I want just really quickly show some demo of how you can use MedSam. At the left is a demo where we can use simple bounding box to do efficient cellular segmentations in pathology images. And the right shows how you can segment the liver and tumor in CT scans just by simple prompts such as bounding box. And you will find that not only MedSam is very fast, but also very accurate in segmenting out small regions of interest, such as organs and tumors. Again, this is a demo on MRI and X-ray. Similarly, you will find that with very approximate bounding box, MedSam can quickly and accurately segment all the organs of interest. We also find that MedSam can very effectively being applied in surgical images. So this demo shows how MedSam can help with surgical segmentations. And we also applied it in surgical videos by simply running MedSam frame by frame. And from the demo we shown here, you kind of see some applications of applying this foundation model MedSam on medical trainings. Since its publication, MedSam has been overwhelmingly received in the community. After its release on preprint, we already got hundreds of citations before publication, also being incorporated in many open source communities such as Hugging Face and Napari, which is a very popular medical imaging viewers. We've also got lots of GitHub code, GitHub development by co-developing these models. So because we see lots of foundation models popping out every day for genomics, such as DNA-BERT, Informer, and including our SCGBD or GInformer, which are foundation model for genomics, we feel it's necessary to provide a review paper so that it can provide educational materials for people who are interested in dive deeper into transformers and foundation models in genomic. So last week, Mika, who is a PhD student in my lab, kind of together we published a review paper in this archive link entitled Two Transformers and a Beyond Large Language Model for the Genome. And in this review, we provide thorough technical survey of a variety of foundation models based on transformers in genomic, such as DNA-BERT, Informer, Big RNA, GInformer, and including our SCGBD as well. So if you're interested, feel free to look at this archive paper and read the review paper. Last, really, despite my personal preference of moving forward with foundation model, I really want to caution that we have to move forward with care because there are many unaddressed problems. For example, we actually don't know how to curate the large-scale pre-training data set beyond single-cell RNA-seq. For example, if we want to develop a foundation model for ATAC-seq, what's the best way to curate this large-scale pre-training data? And second, we really don't have a good way to thoroughly and fairly evaluate a variety of foundation models. So I know many people, many consortium are working towards this goal, but so far we actually don't have a thorough database to fairly evaluate foundation models. Third, how do we make sure foundation models are open and transparent? Because foundation models often are very bulky and very hard to interpret. So how do we develop architect or techniques to interpret, understand these foundation models is very important. Last but not least, these foundation models are very powerful. How do we make sure everyone have access to these foundation models? In other words, how do we democratize these foundation models is also a very important problem. With that, I just want to acknowledge some of the important contributors. SCGBT is mainly contributed by Haotian, Chloe, and Connor. And also Bleep is mostly done by Ronald in collaboration with Sonia and Gary. And MedSam are mostly done by my postdoc, Jing. And we put all the papers and a code database on GitHub. So if you're interested, feel free to dive deeper into these models. With that, I want to conclude the talk and happy to take any questions.
Thank you very much for your wonderful lecture. So there are some questions uploaded in the website. I'd like to read it for you and other people. The first question is that why do you think? Okay. Sorry. Why do you think generative training learn better than purely reconstructing based or random masking based approach like SC formal, considering non-sequential nature of data? That's first question.
Yeah, this is a great question. So SC-Former again was developed by our lab as well before SCGBT. Really, the difference between SC-Former and SCGBT is that SC-Former is an encoder-only architect, and SCGBT is a decoder architect that trained in an autoregressive way. We find that by empirically comparing SCGBT and SC-Former, that generative pre-training can give better embeddings using the same data. And the reason is kind of similar to what we observe in language modeling, that GPT often is more powerful than BERT. We believe that by generative pre-training the data, by predicting next genes, you actually have to understand the whole cell. In other words, by forcing the model to decode out the whole gene expression in an autoregressive way, you're actually forcing the model to learn the underlying data distribution of single cells. So empirically, we also find that SCGBT can outperform many encoder-only models such as SC-Former, SC-BERT, and GeneFormers. So we really believe that autoregressive training can give the model a much better capacity if properly trained.
Okay, I will read the second question. You can also see the questions in your chatting window in the Zoom. The second question is that how does SGPT address highly variable gene selection? Does it need it? Also, if the dataset has different number of genes due to filtering genes, different version of cell ranges, etc., how does SGPT accommodate the issue?
Yeah, very good question. So, in theory, SGPT does not need highly variable gene selections. You can simply just tokenizing all the genes you have. This is the power of foundation model in which you don't need to care about aligning all the genes. However, due to some memory constraint, we does recommend to do some variable gene selection for your own data set, but we test that it's not sensitive to the way you select variable genes. So this is really where we see SGPT is more powerful in some other transfer learning method is that we really don't care about, how many genes you have and how to align genes as pre-processing steps.
Okay. There is the third question. This is the third question. How intensive GPUs do you need to run 3 million cells for pre-training?
I think we trained SCGP using 33 million cells. It took us 20 A100 GPUs, which is one of the most advanced GPUs, and for 2 weeks. So it's very computer heavy process, and also requires lots of kind of caring during such as hyperparameter tuning, things like that.
I think the question is that you use the 30 million, but if we use 3 million, how do you expect the GPU you need?
The 3 million is very lightweighted to train, in the sense that it probably only takes 1 day or 2 day maximum to pre-train SGPT, but the performance is less impressive than training from 33 million cells.
Okay, thank you. The next question is this. For the integration performance metrics, ground truth cell annotations are required. How do you get the ground truth annotations?
This is an excellent question. So we took some benchmark data set with ground truth given, because these are common practice in single cell community. However, we do believe that there's some human bias of the cell type annotations and may not accurately reflect the true cell types. So this is kind of circle back to some of the point I made for limitations of these foundation models, that we don't have really some real ground truth to thoroughly evaluate how good these foundation models are.
Okay. Okay, the next question. Thank you for the interesting lecture. How does SGPT perform on unseen data, such as very rare disease, or can it be applied to other species? Yeah.
This is excellent question. So as you know, SGPT was trained on human normal cells, healthy cells. So it will require fine-tuning if we want to perform transfer learning to diseases. We tested it on cancer. It works quite well just by zero shot on reference mapping, but not on other tasks. For rare disease, we actually didn't have data to validate it yet. We definitely welcome everyone who are interested in testing out to download our model. And if you have data and you want to collaborate, I'm more than happy to really discuss how we can work together to test how generalizable SGPT is for diseases. For other species, it's very hard. So we have some ongoing work to train a foundation model across species. The challenges are data scarcity again, and also it's not very clear to align different genes because the gene names are not even the same across species. So there are lots of challenges, but this is definitely our ongoing work in the lab.
Okay. There are so many questions. We'll continue. The next question is that would the users be able to pre-train the SGPT in their own way, probably in spatial context in the future?
Absolutely. We share all the training code, the model, scripts in our GitHub. So if you're interested, feel free to reach out. If you have any difficulty, running our model, again, let me know how we can help. But we already see many labs that use our SGPT in their own data set and generate some good results. So definitely welcome you to download our model and play it out. Okay.
The next question is this. I would assume that the notion of word ordering in LLM would be a tricky concept to be utilized in SGPT. The genes are words and cells are sentences. How do you handle these features? Do you think relation between genes can also learn in the model?
Yes. So we actually, in the SGPT model, we did lots of experiments on gene regulatory network analysis because it's a natural byproduct for SGPT. Not only we can learn embeddings of cells, we can also learn embeddings of genes. We can actually find lots of interesting gene programs just looking at the embeddings of different genes. In other words, really by predict next important genes, we can actually find the correlations of different gene programs. So this is very exciting for us because it provides a very powerful tool to understand some of the pathways, gene programs, to really interpret how SGBT understands the data set.
Okay, I think this is probably the next question. Regarding your answer to HBG, highly variable genes, could I use additional genes, isoforms, or peaks, even if it may not be in your training data?
That's an excellent question. So in cell by gene, we actually use the whole list of genes, including these isoform or different peaks. Overall, we train on 60,000 gene identifiers in Human Cell Atlas. So overall, I think it's a very thorough list of gene identifiers. So I'm very confident that you can find some correspondence, even if there's some isoforms or some other peaks that we missed, that you can probably simply do a mapping of peaks to the nearby genes. We did that to integrate single cell ataxic. It works quite well. Certainly it's not the best way, but it works empirically very well. Certainly the future direction should be that we can train a thorough foundation model for different kind of biological elements or regulatory elements. But again, as I mentioned, this is some very challenging task because of data limitations.
Okay, another question. What are your efforts to control hallucination, which are quite hard to recognize in gene cell context?
That's an excellent question. So, unlike languages where, like ChatGPT, it hallucinates different reference, for example, it's not clear to me how to understand hallucinations in SCGPT because I think the generative power were underutilized. I think it's a very interesting phenomena to look at, but it's just less straightforward to us. I'm very interested to see how this model can hallucinate because unlike languages, biology contains so many unknown elements. Maybe the understanding the model is hallucinating, maybe some novel biology. Certainly, we have to distinguish between hallucination and error. But I think to me, this is where we can find lots of potentials to find some novel discoveries through these foundation models, such as SGBT. This is also where I think generative pre-training has more potentials than encoder-only models such as SCFormer, in the sense that we can start to look at how SCGB can generate cells, particularly when it comes to novel cell types, some unknown gene programs, things like those. But so far, there's no clear guideline. So this is definitely something we are very interested in understanding SCGBT. But this is an excellent point.
Okay, thank you very much. I think there are more questions, but I think I should move to the next session. So if any of the audience has some more questions, you can contact directly to Dr. Wang or contact us to send the questions. So thank you again, your excellent lecture today, and I move to next. Thank you. You can stay here to see the next lecture. Sure. So okay, I'd like to move to the next one. Thank you.